Okay, taking a look at meiosis. Meiosis is going to result in the formation of gametes. And remember, gametes are the sex cells. When we look at reproduction, we have asexual and sexual reproduction. Asexual was for mitosis. One parent cell produces identical offspring. There's no fusion or combining of gametes. Examples of asexual reproduction: binary fission, budding, and fragmentation. Sexual reproduction involves two parent cells. You have a unique offspring. You have, in other words, genetic variation occurring. And it does involve the fusion of gametes. Organisms that are going to reproduce sexually, they're made up of two different types of cells. The somatic cells, which are body cells, with a diploid number of chromosomes, such as skin cells. Remember, diploid is 2N. Gametes are sex cells with a haploid number of chromosomes. That's going to be the sperm and the egg. The gametes would combine together to make a zygote. Sperm cells are the male gamete. They're produced from the male gonad, the testes. Females produce eggs or ovum. The female gamete is produced by the female gonad, known as the ovaries. When the sperm and the egg combine together, that's going to produce a zygote. During a process known as ovulation, an ovum or egg is released from the ovary and transported to an area where fertilization can occur. The result of fertilization is a fertilized egg known as the zygote. Here we have an egg. Remember, they're going to be haploid. The sperm cell, which is also haploid, they combine together to give us our zygote, which is going to be diploid. Homologous chromosomes. This is when you have a pair of chromosomes, one chromosome from the mom, one from the dad. It's known as the maternal and paternal chromosomes. They're similar in size and shape. The homologous pairs make a tetrads. In other words, there'll be 4 of them. They carry the genes controlling the same inherited traits. Each position on a gene or the locus is the same position on the homologous pair. Humans have 23 pairs of homologous chromosomes. 22 pairs are autosomes or body cells, and 1 pair represents the sex cells. The homologous chromosomes, since a homologous pair consists of 4 chromatids, it's known as a tetrad. Tetrad, if you've ever played the game Tetris, represents the number 4. And looking at this diagram, we have a pair of homologous chromosomes. Notice that the location on the chromosome for the gene for eye color is in the same spot. Again, it doesn't mean that the eye color is the same. It just means that the gene is found on that same location. We have another gene on this chromosome for hair color. Again, it's found in the same area. Humans have 23 sets of homologous chromosomes. Each set of homologous chromosomes consists of 2 homologs. In looking at the homologous chromosomes, the autosomes, meaning body cells, the chromosome sets are going to be 1 through 22. They code for most of the offspring's traits. Sex cells, chromosome set number 23, determine the sex of the offspring. 2 X chromosomes is going to result in a female. An X and a Y chromosome results in a male. The Y chromosome is much smaller than an X chromosome. In looking at meiosis, it's a process in which single cell goes through a nuclear division to produce 4 haploid gametes. Remember, mitosis produce two identical cells. In meiosis, we're going to produce 4 haploid gametes. During meiosis, the 2 end cells are reduced to end cells. So in meiosis, we're going to go from diploid to haploid. If meiosis did not occur, chromosome number would double with each generation and the offspring would die. When we look at egg formation, meiosis in females, remember the female is responsible for producing the egg. One daughter cell keeps all of the cytoplasm. The other 3 cells are going to have genetic material and they're known as polar bodies. Remember, the result of meiosis is producing 4 individual cells. In the case of an egg, you're going to get 1 egg and 3 polar bodies. In looking at sperm formation, meiosis in males, the cytoplasm and the nuclear material divides evenly. The flagella are added last to the cells following the differentiation. So when you have sperm cells being produced, you're going to produce, as a result of going through meiosis, 4 different sperm cells. For meiosis, again, looking at the cell cycle, you have interphase 1. Interphase 1 is similar to interphase in mitosis. The chromosomes are going to replicate in the S phase. The centriole pairs also replicate, and the nucleus and nucleolus are going to be visible. Now we enter into meiosis 1. Remember, there's 2 divisions in meiosis, meiosis 1 and meiosis 2. Meiosis 1 is characterized by the separation of the homologous pairs of chromosomes. There's 4 stages for meiosis 1: prophase 1, metaphase 1, anaphase 1, and telophase 1. Notice we still have the P map, but now it's followed by the number 1, indicating that we're looking at the first part of the meiosis process. In prophase 1, chromosomes condense, the nuclear envelope disappears, spindle fibers form, synapses occur, the homologous chromosome pairs up to form a tetrad. A tetrad is 2 chromosomes or 4 chromatids. Crossing over can occur. If you're looking at the tetrad here, you'll notice you see the 2 sets of chromosomes. You have 2 sets of pairs here. As we go through the next few slides, make sure that you're also watching the colors on the chromosomes, that dark blue versus the light blue. When we look at our homologous chromosomes, one of the chromosomes, just to help orient us, is the darker blue, and then a second one is the lighter blue. And so our pair of homologous chromosomes, we have the dark blue one and a light blue one. It's possible to have crossing over. Segments of non-sister chromatids break and exchange genetic material. This is going to allow for a greater genetic diversity in the offspring. Here we have non-sister chromatids. Notice one's the dark blue, one is the light blue. When crossing over occurs, you see where the dark blue is overlapping with a segment of that light blue. And as a result, the segments are going to break off and combine with the other one. So as we look at our tetrad again, notice that our dark blue chromosome has a segment at the bottom right that's also now a light blue chromosome. And the segment of that dark chromosome, the dark blue one, that segment has now been moved to the light one. Again, this is where you have the chromosomes are so close together that the sister chromatids can actually cross over one or the other. And as a result, pieces break off but recombine, but with a different chromatid. We now move to metaphase 1. The homologous chromosomes are going to move to the middle, again at the equator, as tetrads. Here we can have independent assortment occurring. The orientation of the homologous pairs to the poles is random. If we look at this first setup, we can have a pair of chromosomes. Again, you're looking at the colors here. Notice the difference in the colors of the chromosome pairs and how they've lined up versus the second picture. Again, looking at the coloring of them. Notice that on the left-hand side in the first diagram, that bottom chromosome is a dark blue. But on the right-hand side, the bottom chromosome on the left-hand side is a light blue. Because as you move to anaphase, the chromosome pairs are going to separate. And so it's important to note that the independent assortment, again, is going to allow for a greater genetic diversity. In anaphase 1, the homologous pairs, they separate and move to opposite poles. The chromatids did not separate from the centromere, it's just the pairs are separating. And so based on how they're separating, again, this leads to a greater genetic diversity. We now move to telophase 1. The nuclear envelope usually does not form. Chromosome number in 2 new cells is now half because the chromatids did not separate. Cytokinesis may or may not occur. So you'll notice here, again, you see the pinching beginning, but it may not completely separate. When we look at the cells that are forming, notice we still have those chromosomes. That brings us to meiosis 2. Remember, there's 2 cell divisions in meiosis. So now we're going to look at meiosis 2. There isn't any replication occurring of the chromosomes, but it proceeds like mitosis. It starts and ends with haploid cells. Here again we have our chromosomes for prophase 2. Remember, now we're in meiosis 2, so prophase 2. Moving to metaphase 2, and you can notice where that line cuts through the middle of the chromosome. Now we are going to separate in anaphase 2, separate those chromosomes, separating the centromere. Here's anaphase 2, where the chromatids are now separated from one another, again splitting that centromere. Telophase 2, where we have the pinching in occurring for each of those cells, resulting in cytokinesis of 4 new daughter cells. And then the 4 new daughter cells are going to have different genetic material, in other words, different chromosomes, depending on how the chromosomes lined up.